Hi, I'm Rithik Raya from Pineview School for the Gifted, and today we've got someone whose work literally connects physics, biology, and data science, Dr. John Quackenbush, professor at Harvard and the Dana-Farber Cancer Institute, and director of the Center for Cancer Computational Biology. And he's one of the pioneers who helped turn biology into a data-driven science, and he used that knowledge to understand and fight cancer. Dr. Quackenbush, it's great to have you here.
Very nice to be here with you today, too.
Yes. Okay. So you've had such a wild career path. You went from theoretical physics and then to genomics. So let's start there. You started out studying theoretical physics and you started leading the, I believe, Human Genome Project. What made you switch from like black holes to biology?
Well, I didn't lead the Human Genome Project, and I wasn't working on black holes. I was actually working on something called string theory. But the things that happen in your life um, often are things you don't really prepare for. And I studied physics, and I thought this is what I wanted to do. I really loved the field, but when I was finishing my PhD, it was actually a bad time to be doing physics here in the US.
Okay.
We had a real reduction in the funding for physics research, and that was driven in part by the end of the Cold War. So history sort of stepped in and played an interesting role. Physicists had long, since the Manhattan Project in World War II, it had talked about the importance of um, having physics research funded in the US because if we didn't, our adversaries would get ahead of us with potential weapons development. And it was sort of an disingenuous way of justifying doing physics because nobody I knew actually worked on weapons research, right? We were all doing studies in fundamental physics, trying to understand what makes the universe tick the way it does. But when the Cold War ended, funding for physics research in the US started to evaporate. So you may know, or may have heard, maybe not, of something called the superconducting supercollider. And this was going to be built in Texas. So the way physicists learn about fundamental interactions in nature is they generate very high energies by taking particles at high energies and smashing them together. And it creates conditions that are very similar to the conditions that existed in the early days of the universe. So, by smashing these particles together, we can actually probe small distances, but also probe interactions between particles. And there's a big collider, big particle physics laboratory in Europe called CERN.
Mhm.
There was a slightly smaller one here outside of Chicago called Fermilab, and they were going to build this big accelerator under the plains of Texas. And it was huge. It was going to be 54 miles around.
Wow.
And they dug about 18 miles of the tunnel, and then funding got cut off because it just wasn't a priority given other things. And in a lot of ways, that was really unfortunate. But personally, what that meant was that the funding that was supporting me evaporated. So I had a job as a postdoctoral fellow. So working for a few years after my PhD, I had that job lined up to work with one of the best theoretical physics groups in the US. Their funding got cut. They didn't have anything else for me to do, or no way to pay me, at least. They had a lot of things they wanted me to do, but it's hard to work when you don't have food and rent. So I had to think about what I would do next or what else I would do. And I was fortunate that an experimental physics group at UCLA, where I'd done my PhD, offered me a position. I worked with them for 2 years. I was doing experiments at Fermilab. We made proposals that were actually accepted to look for things like antiproton decay. We wrote another proposal to build a dedicated electron-positron collider at UCLA in one of the parking lots. Um, and I was really excited about the work I was doing, but after 2 years, the funding disappeared. So I was sort of stuck and I had to figure out what I was going to do next. And I was fortunate in a lot of ways. My wife always jokes when I tell this story that I shouldn't be saying this, but um, Part of the reason I ended up in the biological sciences was I had a girlfriend who was a biologist, and I used to hang out in her lab late at night when she was doing work on, believe it or not, hormone regulation of gene expression in cockroaches. And I would hang out with her and I'd start to help her and her friends analyze data. I got very interested in all this biology that had been invented since I had taken my last biology class in high school. And so I had this opportunity to sort of jump into some seminars and working groups and listen to what people were doing. And right around that time, the Human Genome Project was just getting off the ground. And if you want to hear the history of the Human Genome Project, I have some very interesting stories about that
Okay.
too. But the Human Genome Project was just getting off the ground. And believe it or not, had started in the Department of Energy. We can talk about that. But it was taken over by the National Institutes of Health, the NIH,
Mhm.
and they recognized they needed people with quantitative skills to work on the Genome Project. And so I'd been helping my girlfriend and her friends analyze data and learning biology. So I applied for something they just started. It was called the Career Development Award. It was a 5-year fellowship, a grant to work on the Human Genome Project. And believe it or not, I was one of 3 people who applied in the very first round. There were 2 computer scientists and some other guy, and they ended up picking one of the really good computer scientists and the other guy, and I just happened to be the other guy, the physicist. And so I started down my path on the Human Genome Project.
That's fascinating. So, like, do you think physics, gave you the ability to see biology as like a network of interacting systems in the project?
So, I think my training in physics, your training in almost any discipline, does 2 things. It gives you a set of tools, right? So, like, I haven't solved the Dirac equation in more than 30 years, but I used to know how to do that. I used to do it all the time, right? So, you have these tools, these things that you know how to do. But fundamentally, if your education is doing the right thing, it's teaching you something else. It's teaching you how to ask questions. And so you start to think about problems. You start to think, okay, how does this work? How do I tease this apart? What are the fundamental questions that are unanswered when I look at the system? And that's really what physics, my education gave me the ability to do is to ask questions. The nice thing coming into a field from the outside too is that you have some latitude to ask the stupid questions, right? The things that everyone knows. And what you discover pretty quickly is what everyone knows is often not known by very many people, if anyone. So you can ask questions, and someone will say, "Oh, I don't know," and that's been very helpful too. But to your question about how my physics education in complex systems works, I think one of the things that I've been good about doing over the years is being able to sort of step back and use analogies from the things I've learned in physics as a way of trying to tackle some of the complex problems in biological systems.
Okay. So you think like that cross-field mindset is what biology needs more of now as well?
You know, I think people coming in from different disciplines can contribute. You also see people come in and think that biology is physics and it's not, or biology is computer science and it's not. Um, to really be effective, you can't just stand on the outside. You have to become part of the ecosystem and you have to understand the problems the really pressing problems in the field. And then you have to think carefully about how you're going to go about tackling those problems, right? What are the right questions to ask? What are the right tools? What are the things we can do? And what are the things that simply we're not going to be able to do? And those are all really dependent on understanding the systems, right?
Yeah.
You see a lot of people jumping to, machine learning and artificial intelligence, and they say, "Oh, it's going to solve biology." At the end of the day, most of those ideas don't work because they're not really grounded in our understanding of biological systems. And that's more important than you might think.
Wow, that's crazy. So, you also mentioned, like in the previous question, that you worked at the Human Genome Project and that it was part of the Department of Energy. Could you like clarify on that?
Sure. So the Department of Energy, believe it or not, funded most of the physics research in the US. And they ran the big national labs like Fermilab that I mentioned and a few others. It was really the Department of Energy that was driving physics research forward. When funding for physics started to be cut by the Congress, a number of people at the Department of Energy sort of stood back and said, all right, if we can't do, our big physics, what's the next big question that we can tackle? And it turns out they leapt to the idea of sequencing the entire genome. So when I say sequence, DNA is a long chain polymer. It's 3 billion bases in length, right? That's a number which is, your genome is 3 billion bases in length.
Yeah.
That's a number which is difficult for me to comprehend, right? It's slightly smaller than my salary, tiny fraction of the national debt. But 3 billion is the number of seconds in 95 years. So that gives you some sense of the scope and scale. And you actually have two copies of the genome in every one of your cells. So 6 billion bases of DNA. So reading that off presented some technical, computational, conceptual challenges. And so the Department of Energy said, well, let's do it. We're the big science people, right? A lot of the NIH funded research was gene by gene, trait by trait. The Department of Energy said, let's just do it. Now, you might think, well, why is the Department of Energy even thinking about genetics? And it turns out, believe it or not, that history is really important. And the Department of Energy, if you look at its history, it grew out of something called the Atomic Energy Commission.
Oh,
And part of their mandate in the early days was to understand the effects of ionizing radiation, right? Because if you're doing atomic energy, you have radiation, you have to figure out what it's going to do to humans. And so um, they had a long history of funding research in genetics. And they said, well, we can do big science. Let's do the biggest genetic experiment we can do and sequence the genome. So there's actually a conference that was held in the Silmar where the Department of Energy brought all these people together to think about and plan out how they're going to do it. Pretty quickly, the National Institutes of Health took over, which probably made more sense. But the DOE still played a really major role throughout the sequencing of the genome in helping to really dictate the strategy to push technology forward. So, they're really big players early on.
okay.
But, that's sort of how how the genome got started, the sequencing of the genome and the Human Genome Project got started in a place you probably wouldn't have expected.
Also, speaking about the Human Genome Project, like you mentioned that you had some like funny war stories, let's say. What was it like on the inside?
Well, the Genome Project wasn't just one thing, right? The human genome was sequenced by groups distributed around the world. And the biggest concentration of laboratories was probably here in the US. There are quite a few in Europe. There were some in Japan. In Europe, it was a little bit more dispersed too. The biggest sequencing center in Europe was actually in the United Kingdom, the UK, at a place called the Sanger Center, which was built just outside of Cambridge. So there was a big sequencing facility there. There were a few in the US. There were some smaller sequencing labs. And it was really a collective effort to sequence the genome with public funding. What happened at some point was a gentleman named Craig Venter had been at the National Institutes of Health and got involved in sequencing human genes. And he eventually got involved with a company that got spun out, Human Genome Sciences, and used some of the money that was dedicated to launching Human Genome Sciences to build a nonprofit research institute called Tiger, the Institute for Genomic Research. And Tiger was actually a major player in sequencing with government-funded uh, grants to sequence human and other genomes. But what happened was that Venter, after a few years of being involved in the Human Genome Project, got recruited to help start another company called Celera Genomics, which was tied up with one of the manufacturers of the instruments that was used um, to sequence the genome itself. So we had this really interesting time when you had this public project racing to sequence the genome and this private company, Celera Genomics, racing. And the two of them um, were using slightly different strategies, but really not that different overall. Very complementary to each other. But it was this kind of race between the public and private sectors. And, it was always called a race, but it's hard to imagine a race because the public project was committed to releasing its data weekly, and the private project wasn't committed to releasing data at all. So when you have your own data and the public data, by definition, you have more, right? So they were always sort of ahead. Nevertheless, um, in the early 2000s and about 2005, no, I'm sorry. I have to think back now. I have to really think about the history. In 2000, um, the public and private projects were really battling with each other. And at some point, believe it or not, people from the Department of Energy stepped in and kind of brokered a truce where they announced that the genome was finished. And then within a few days of each other, one in the scientific journal called Science, the other in the journal called Nature, published their analysis of the human genome. And the public project was a few days ahead, I think by design, but they essentially announced at the same time the genome was finished. So was it finished? Well, yes and no. It was actually refined over a long period of time and only a few years ago announced completely finished, even though there's still some gaps. But it was finished enough that we had a pretty good idea of the organization of the genome. We had a pretty good catalog of the approximately 25,000 genes encoded in the genome. And we really had a blueprint that we could use to springboard our understanding of human health and disease.
That's really cool. So, like, did you realize, like, when you first started that you would be part of something that was redefining medicine, like, as you know it today?
So, it was an exciting project that had a tremendous amount of potential. And it, you're asking me earlier about complexity. Understanding the genome really speaks to, on multiple levels, speaks to the need to embrace the complexity of biological systems. So, the easiest way for me to start this discussion is, to look at you and to look at me, right? My skin is pretty pale, yours is darker, right? I have light eyes, you have pretty dark eyes. My hair, you know, it's a little wet right now because, like I said, I just got out of the shower, but, it didn't used to be white. It used to be at least brown, right? Yours is pretty dark, right? These are traits that are controlled by some of our genes. But if I look across that 3 billion bases in one copy of our genome, the difference between your genome and my genome is about 1/10 of 1%,
Wow.
right? So one base out of 1,000. And most of those aren't in the genes. There are about 25,000 genes, and most of the genome is not genes. So they're really minor differences. But, if you think about 6 billion bases and the two copies of the genome we carry, that means there are about 6 million differences between your genome and my genome, right? So we suddenly have a really big combinatorial problem we have to start thinking through if we want to think about traits, right? And eye color, hair color, skin color, they're not that important to really think about in the long term because, at a fundamental level, you and I are more alike than we're different. Okay. So, we eat the same or can eat the same food, breathe the same air, right? Uh, any male and female of reproductive age can potentially produce a child, right? I can give you a blood transfusion. I have O negative blood because I'm a very giving person. It's a universal donor. But no matter what your blood type is, you can take my blood and it will circulate and function perfectly, right? And at these fundamental levels, we're just not that different from each other. But some of those small differences influence the expression of traits, including our risk of developing diseases, in part how we age, right? And so teasing apart what these differences mean has really been, I think, part of the exciting challenge. And it really does involve embracing the complexity of biological systems. And I'll come back to that later, but yeah, there's a lot more to think about as you try to tease this apart.
That's crazy, yeah. So, I did my research, and you mentioned that you were on the same team with the computer scientist, and I found out that what you guys were doing was called precision medicine, and you were building the foundation for it. Did people back then even believe that computers could actually help decode life?
Well, I think from the beginning, we realized that we couldn't sequence the genome without the involvement of computer scientists, right? That computational systems are going to be necessary. We talked about the size of the genome, right? 3 billion bases, the number of seconds in 95 years. Imagine smashing that into millions and millions of pieces and then trying to put it back together, right? That's a combinatorially difficult problem to try to solve. If you then start to think about 25,000 genes, but millions of genetic variants, and to really understand how those genetic variants and those genes come together to influence our traits, right? That's not the kind of problem you can solve using an abacus or, a piece of paper and a pencil. It's a difficult challenge where the combinations and permutations of things that can come together to influence traits really becomes overwhelming unless you have the tools to start to tease through large quantities of data. So I think we've always recognized from the beginning that computational methods and quantitative approaches were really going to be at the heart of our journey to understand how our genes translate into the traits that we manifest.
That's so cool, yeah. So one of the things that you really championed is like systems biology, the idea that we can't understand disease by looking at single genes. And I heard you mention that before, like the traits and stuff, but also for diseases. When did it hit you that like data and computation could reveal like those patterns that biology alone couldn't?
Well, I would say biology alone can't do it, but I think a lot of science, the way we approach science, is very reductionist, right? We take large complex systems and we break them down to their simplest components, and we try to understand those. And we really run the risk of oversimplifying the systems that we're studying. Often in physics, it's very good to reduce things and then build them back up. But we have what we call first principles that we can fall back on. They're basic um, reflections of our understanding of how the universe works. Things like symmetries, right? Um, Translational symmetry, the fact that physics looks the same. Where do you live?
Florida.
Florida, okay? So physics in Florida and physics in Boston are going to be there, and Massachusetts are going to be the same, right? And the fact that we can move an experiment from here to there and get the same answer, that simple translational symmetry is what gives us conservation of momentum, right? The fact that I can do an experiment today and tomorrow, right? This time translation symmetry get the same answer. That gives us conservation of energy, right? There are all these different pieces in physics that we can fall back on. In biological systems, we aren't at the point yet where we understand them well enough. And they may simply be too complex for us to ever get that kind of understanding, right? Those first principles that we can build theoretical models on top of. So, what we really have to do is we have to rely in large part on our understanding of biological systems as we start to tease out what drives things like health and disease, right? We have to understand evolutionary processes and how they made a system that works well, but may not be optimal, right?
Yeah.
Um, and you know, really teasing out the complexity is something I think I've embraced almost from the beginning, right? Once I started looking at biological systems and started thinking through
Hmm.
the challenges of making sense of all of this, that you realize that you have to embrace this kind of complexity, right? So the easiest way for me to sort of explain this is to say, you know, we all know, right? We're talking about your eye color, my eye color. We all know that those traits have a genetic component, right?
Yeah.
Like there's no chance I could ever have, well, maybe if I had a wife with really dark eyes, you know, but not a great chance that I could produce with another blue-eyed spouse, a child with brown eyes, right? We know that our traits to a certain extent are affected by our genes. Now, when I say eye color, that actually opens up another door because many traits like eye color, like skin color, they're polygenic. They're influenced by a number of different genes and gene variants working together. But if we set that aside, right, think about this, right? Genes may influence our traits, but every cell in your body has more or less the same genome. Okay. There might be a few variants, but overall, it's the same genome,
Yeah.
right? What does the structure of a brain cell look like?
Like a dendrite and then the axon.
Right. Well, you sort of have this star-like
Yeah.
cell body with the long dendrites and or long axons and dendrites, right? So they can reach out and connect to other brain cells,
Hmm.
right? And they're making neurotransmitters. If you look at a liver cell, they're small and compact,
Yeah.
right? Hopefully they're not making neurotransmitters, they're making digestive enzymes, right?
Yes.
Their morphology, right? How they're constructed physically, their their traits, one's making neurotransmitters, the other's making digestive enzymes. They're very different. Now, some things they all have to do, right? They all have to consume oxygen, produce carbon dioxide, burn sugars, replicate, but at a really profound level, their shape and function is different, yet they have exactly the same genome. And that should be the first clue that what's encoded in our genome is only part of the story of what makes us who we are.
So like the grammar behind the DNA, in essence.
Well, there's grammar, but there are also many different factors that influence how our traits manifest. So I tend to think of these as control systems, right?
Hmm.
They're governing what genes get turned on and off, and they're governing when and where those genes are turned on and off. So, during development, right, you start off with a fertilized egg, which is basically a blank slate, right? Because that egg cell doesn't, it's not ever going to be a brain cell, it's not going to be a liver cell, but it starts to divide. And as it divides, its progeny start to take on different traits, right? So we call it a pluripotent stem cell, right? It can become anything. But over time, those traits start to change, right? The way they manifest starts to change. And we think of that as being part of a regulatory process, right? Things are being turned on and off. So just like you can go, to an electronics store and buy a bunch of, electronic components. Right? You could maybe build a radio, you could build an AM radio or an FM radio, and the parts are going to be more or less the same, but they're going to be some that are fundamentally different, right?
Yep.
It's the same sort of thing. You've got a lot of parts that are used ubiquitously in all the different cell types, but then you also have some that are put together differently. And they're regulated differently, right? When the genes are turned on and off during development, but also in response to different perturbations, that is all regulated by a set of complex processes. And some of these are inherent to the cells. Some of them actually reflect things like environmental exposures that change what we call the epigenetics, right? So you've got these systems that are adapting to stimuli from their environment, that are recording effects of things like environmental exposure, but then also are fundamentally designed to regulate processes. And that regulation is really important for understanding a whole host of questions in health and disease.
That's so cool. Yeah, definitely. The variety of factors and everything. Like, it's kind of like the nurture versus nature argument, but like amplified with more.
Well, nature's going to play a role in all sorts of things, right? If I walk out of here and fall down the steps and hit my head, right, nature is going to determine the length of my lifetime potentially far more than well, I don't know if that's nurture or nature. Maybe that's nurture, but it's really our environment. Our genome manifests itself by interacting with its environment, but some of that environment is actually encoded within the genome. So there are external forces and internal forces that all work together to make us who we are.
That's, yeah, very interesting. Yeah. So, do you think like biology is ever going to turn into a branch of data science?
I always like to say that the winners and losers in the race to understand human health and disease are going to be those who are best able to collect, manage, analyze, and interpret data. So, I think data science is always going to play a big role. One of the other things I'd like to point out to, my students and postdoctoral fellows
Yeah.
is that any experiment you do produces one and only one thing, and that's data. And sometimes the data is well collected, the experiments are well designed, sometimes they're not. But, you know, these experiments are producing data. And so the way we advance our understanding of biological systems is by doing experiments where we collect the right type of data, but then we have to analyze it and interpret it. The, the sort of corollary to that is that if we look at just taking, data science, everybody gets very excited about machine learning. Oh, machine learning is going to solve human health and disease. And the truth is, it's just not. Machine learning methods are very good at finding correlations, but they're not very good at giving us information about how and why biological systems function the way they do. So that sort of causal link between our observations and the traits that manifest is one that's much harder to establish. And, if you think about 25,000 genes encoded in the genome and all these other factors that are influencing how these genes manifest the traits that we observe, you pretty quickly recognize that it's not a simple problem, right? It's not like looking at all the movies you watch and recommending something on Netflix, right? Where, oh yeah, we're going to say, you'll like this film. First, you know, the data is more complex than just a few things you watch. Second, if you're a patient and I'm a physician and I'm treating you, right? And I'm trying to do, we mentioned precision medicine. I guess we didn't talk about it very much before, but if we, think about trying to uh, specifically define how you're going to be treated if you had a particular disease, based on your own genetic background and other factors, then, you know, if I get it wrong with you, that's bad, right? If I give you a
Yeah.
drug that kills you, that's bad. If I recommend a movie you don't like, nobody cares, right? You watch 5 minutes, shut it off and watch something else. So the stakes are pretty high. But, one of the things that we can do to improve the ability of these computational systems to, one of our cats is coming to say hello. This is Chloe, by the way. So she'll probably try to crawl up on me.
She's
But,
adjusting.
one of the things that we can do is try to guide those systems that we're going to use, these computational systems and tools, we can try to guide them by using some knowledge of the biological systems that we really want to understand. So, in the work that I and my colleagues have done over many years, um, what we've really tried to do is to bring in a framework that reflects the properties of the biological systems we're trying to model, right? It's not just, the big linear algebra machine and machine learning, but it's really um, a set of properties around which we try to build our models and optimize them. And, the heart of this is really an idea that comes out of computer science. There's a brilliant paper in 1997 from two guys, Wilbert and McCready, who at the time were working at Microsoft Research. And they wrote a paper on something called no free lunch theorems for optimization. And it's a really nice paper, but at its core is this basic idea that there's no computational system that performs optimally in all situations. And so if you want to try to, it was all about optimization. If you want to try to find something which optimizes any model, they argue very persuasively in this paper, the best way to achieve that is to start with a guess that reflects your knowledge of the system. And so really what we try to do is we try to use all these sources of information to try to guess what the structure of regulatory networks in cells look like, and then optimize them so that I can look at your liver and my liver, or
Yeah.
healthy cells and disease cells in my liver, right? To understand how these networks are changing between you and me and changing between my healthy cells and cells that are manifesting disease traits.
Okay. Yeah, that's really amazing. Like turning data into more easy ways to comprehend. Yeah.
Right. And it's
So,
sort of using what you know to guide things,
yeah.
right? So I don't know what classes you're taking now. My PhD was in physics, and, you know, you can even see things in very elementary problems, right? Where, yeah, this won't be using the no free lunch theorem, but it's just kind of building your intuition in, right? If I throw a ball out of my window here, which is on the second story of our house,
Hmm.
it's going to fall down and hit the ground, right? And just my physical understanding and intuition from living, I won't tell you, it's almost 64 years. All right, my own physical understanding will tell me that it's going to hit the ground in a second or two. Right? If I try to calculate how long it takes to land and get 200 seconds, I know something's wrong. Right? When I used to teach physics, students would come in and say, "Oh, I plugged the number into my calculator and this is what came out." And I'd just say, "It's wrong." But how am I supposed to know? My calculator told me. I said, if it's telling you it's going to take 200 seconds to fall 30 feet, right? A ball, that's wrong. So building in your intuition can be an important check on how you model systems and how you understand the results of the modeling that you're doing.
Speaking of modeling, your current focus at Dana-Farber is on cancer genomics, modeling how tumors evolve and resist treatment. When you look at cancer through your experienced lens, what's something that the public doesn't usually understand?
I think there are quite a few things. I'm really now based at the School of Public Health. I have affiliations at Dana-Farber and also at Brigham and Women's Hospital. A lot of my work has been in cancer, but also in pulmonary disease, lung disease.
Hmm.
But, you know, to your question, I think a lot of people think that there's an easy cure for cancer. And cancer, the challenge is that cancer is a lot of different diseases. It's not just one. The other thing that's important to understand is that the individual cells in a tumor are all trying to do the same thing you and I are trying to do, right? Which is survive. And so, you know, as cells in a tumor are growing, they're adapting. As cells are dividing, they're adapting. And one of the things that makes them very adaptable is the fact that one of the processes that breaks down as cancers grow and develop is DNA repair and DNA editing. So there are a whole set of systems that evolutionarily have come about to keep cell replication of DNA as high fidelity as it can be, right? You don't want to introduce errors. Those mechanisms break down in cancer. And so cancer genomes, as the cells divide, start to pick up many, many, many different mutations. And just like we talk about natural selection, right? Survival of the fittest. You have to survive long enough to produce offspring. And in the case of cells, it's long enough to divide. But if you have cells that if I'm giving someone chemotherapy, if they have cells that pick up a mutation that allows them to better survive the chemotherapy, other cells may die, but the ones that can make it through get a big selective advantage. They grow and they propagate because they're not being killed, right? And so tumor cells really do try to evolve and adapt. So, you know, I think it's important to understand that the problem of treating cancer, I'm not going to say curing, but the problem of treating cancer is really complex. We have a great set of tools. We have drugs that, allow 90% of patients to live in many cancers now, where 10% don't. But, 50 years ago, 10% lived and 90% didn't. So we've made huge strides forward, but we still have a lot of work to do, and there's still um, a lot to discover because it really is such a complex disease. Every time someone comes out and says, "Oh, there's a miracle cure," yeah, it's a miracle until someone relapses.
Yeah.
And that happens just because the cells that are best able to survive are the ones that do survive and come back. The other thing that I find sort of disappointing is people say, well, you guys doing cancer research, you know how to cure cancer, you're just hiding it for some reason. First, if that were true, I would take that idea and I'd be all over, TV, the media, social media, wherever it is, touting that idea because if it really worked, I would be a trillionaire, right? If somebody knew how to cure cancer, they'd step up and do it, right? Not just because I'd be incredibly wealthy, but there's yeah, an ethical
Obligation.
imperative to, make things like that known. Um, we're getting much better, but we still have a lot of work to do. And, we talk about disease, but it's really part of the larger question, right? What makes us who we are? How do our traits manifest or how do our genes manifest into the traits that really make us all unique and distinct? And how can we understand how this process, these gene regulatory networks, break down as diseases develop and progress? Because at the end of the day, answering those questions is going to allow us to treat disease, but it's also going to allow us to understand human development and human health far better than we do today.
That's intense. Like, cancer is constantly adapting to survive. Dr. Quackenbush, you've co-founded Genospace, which takes genomic data and makes it usable in hospitals. What made you want to step out of like the academia and like research and start a company?
So it's sort of an interesting question. Academics aren't always as altruistic as we might hope they are. We actually started trying to build systems that would collect large amounts of clinical data at the Harvard hospitals and make it available for research purposes. And for a variety of reasons, people were very hesitant to make those data available. So a colleague and I, Mick Carell, started thinking about the fact that um, yeah, there might be places, hospital systems, disease foundations, others that would be interested in investing in buying a system or having a system that would allow them to really integrate. At the time, what we were trying to do is clinical and genomic data together in a common warehouse to really give us new insights into disease processes. So when we started to run into opposition at the Harvard hospitals, and this was 2010, 2011, we realized that we could just sort of step outside of what we were doing and launch a company.
Yeah.
So it was a very interesting time. Um, we had a lot of ups and downs, but we formally launched the company in about 2012. And then in 2017, we sold it because we were able to really solve the problem of matching um, patients in hospitals where they've been genetically or genomically profiled to look for their cancer-associated mutations to match those patients to ongoing clinical trials that we're often developing drugs looking for particular patterns of mutation as criteria for including patients in the study. So we came up with a way based on all of our understanding of doing that kind of mapping very quickly and very efficiently. And it was probably the fourth or fifth thing we tried, but at the end of the day, that's what let us sell the company.
So that's amazing. Yeah, turning massive data into something like even doctors can use right away. So,
Well, we're doing that now. So 2 summers ago, I had two high school students work with one of my postdoctoral fellows, and we built a data repository that we call Seahorse. So my group actually maintains, I don't know, about 20 different software tools for inferring and analyzing gene regulatory networks. So we have this large collection of tools. The very first one we built was called Panda. And I can't tell you what the acronym stands for, although N is for networks and D is for data. Um, but, why do we name it Panda? Exactly, because pandas are the cutest animals in the world. But, if you name something after cute animal, people remember it. That we had condor, we had lioness, we had a whole set. So now we have about 20 different methods, all named more or less for animals. There's dragon and phoenix and a couple other things, monster. And I really want chupacabra one day. But we built this resource we called Seahorse, and serendipity is luck. So we call it a serendipity engine. And what we did was we said, look, we have these big data sets. Computation is cheap. What we really do in analyzing data is we look for correlations between different data elements. So, and we want to take them farther along the road to causality. But, at the end of the day, we're really looking for things that are associated with each other. Computation is cheap. If you have big data sets, why not just find the correlations between everything and everything else? And so these two high school students looked at some large public data repositories and just did the all by all calculations
yeah.
and put them in a database that you can query. And it's really interesting when you look at physical traits in the one data set we started with, something called GTEx, which has gene expression data in, I think, 38 different distinct tissues. if we look at that, and this is in almost 900 individuals, if you look at that data, what you discover
Yeah.
is that things like height are correlated with some very interesting things. So, one of them is in skeletal muscle, height
Hmm.
is correlated with the expression of genes that are associated with catabolic metabolism, right?
Oh,
Which is interesting because you think, well, maybe tall people just metabolize things differently. That's how they become taller. But in, I think it was 21 different tissues, we saw that height was correlated with the expression of genes that we tend to associate with cancer. Now, that's really odd.
yeah.
But if you look at, and it's something I never would have ever expected, but if you look at the epidemiological literature, there is actually some support for the idea that taller people are at greater risk of developing some cancers. And in fact, all the cancers that we found where alterations of cancer-associated gene expression occurred were all in tissues that had been reported to have some association with height. So being tall doesn't cause cancer, but some of the processes that are involved with being tall may alter our risk of developing cancer. And, things like that are things you don't expect to find, but you can find if you use these large public data sets.
That's really inspiring, yeah. Like unexpected connections. So, if
Exactly.
you look ahead 10 or 20 years, what's the frontier for genomics?
Yeah. First, I don't think I'm going to be out of a job anytime soon. I think there's still going to be open questions. And I think as we look at how we treat disease, we're going to continue to be more intelligent about how we do that. In genomics, transcriptomics, so looking at the genes that are transcribed, genes that are turned on, is important now. It's going to be increasingly important. Epigenomics, so the epigenome, epigenetic effects, are those effects largely that come from environmental factors, although some come with time? Um, these are factors that influence how our genes are expressed, what turn them on and off. That's going to become increasingly important. And just our understanding of how disease processes um, change and alter over time are things where they're going to continue to be open questions, no matter how long we pursue them. But for me, it's just so exciting to be at a place where we have the kind of data that we can use to ask these questions. We have the computational tools and resources to try to answer them. And we have the opportunity to really look at the complexity of biological systems and appreciate that complexity not as a barrier to understanding, but actually as a gateway to teasing out the processes that do alter health and disease. So, one of the things I'll leave you to think about, which is interesting, is a lot of diseases. We talked a little bit about cancer. I also work on lung diseases. And
Hmm.
one of the diseases we study is COPD, chronic obstructive pulmonary disease. And when you look at the phenotype, right, the physical manifestation of traits, COPD looks like a process of accelerated aging in the lung because we all lose lung function as we get older, but in people with diseases like COPD, it just happens much faster.
Yeah.
What we're starting to see is that some of the epigenetic factors that we associate with just regular aging also happen much faster to a molecular level in COPD. So these epigenetic factors are actually changing in ways that alter the manifestation of traits, right? And
Yep.
so that's something new that we're just starting to explore. And I think as you and your colleagues enter the sciences and start to contribute, right?
Hmm.
These kinds of questions are going to still be there, right? And we're seeing all sorts of things in solid tumors in males. Male tumors lose the Y chromosome. And in biology, they tell you the Y doesn't do very much except maybe carry something called testes determining factor. But other than that, whether or not you have testicles, the Y chromosome, that's sort of it. There are very few genes on the Y chromosome, but they seem to play important roles. And one of the things we're seeing is male tumors lose Y chromosomes. As they do, through a series of interactions, it appears they're better able to hide out from your own immune system. So your body can't kill those cells because losing the Y helps them hide, right? And this is just very interesting. One of the other things we invest a lot of time studying is just how an individual's sex influences their disease risk, disease development, disease progression.
Yeah.
Females, biological females, have twice the lifetime risk of males in developing Alzheimer's disease, right? Even correcting for the fact that females tend to outlive males. In colon cancer, males are at a much greater risk of developing colon cancer, but they respond better to chemotherapy. And those are some of the problems we're starting to tackle and tease out too. So they're just, the more things we learn, the more questions we have to answer. And so I think there's great opportunities for people like you who have a solid background in the biological sciences, who also have good quantitative skills to step into the field and really make important contributions.
Well, yeah, I 100% agree. I just wanted to say, Dr. Quackenbush, this was incredible. We went from physics to cancer to starting a company all in one conversation. Thank you for sharing your story and your wisdom, and I think a lot of young people are going to find this super inspiring.
Well, I hope so. My hair's dried a little bit. Um, I was boxing earlier, and I box and I do taekwondo. I was boxing earlier, but I okay, call them back. Um, and came home, did a lot of chores, but I was able to get ready for this and have a great conversation with you. So I do hope people find it inspiring, and I hope they take this as an opportunity to really think about, what their futures are going to be. You know, my advice to you is, try to get good quantitative skills, try to get good programming skills, but really follow your path and follow the things that you love. And, if obstacles get thrown in the way, try to look at those obstacles as opportunities to do something new and exciting.
Thank you. Well, yeah, I appreciate it.
And, good luck with your college applications. My son's now a sophomore at University of Chicago. He went through that a little while ago, and it was just an incredibly painful process. So good luck and don't feel bad. It's such a lottery these days. But, if you're listening to this podcast, you're probably thinking about science. You're probably in a good place to move forward.
Well, yeah, thank you again.